Today I am discussing about the cytoarchitectural organization of the cerebral cortex. Here is a picture of Konstantin von Economo. He's an Austrian. He was a pilot. From engineering to medicine, his flight was. And he painfully mapped the entire cerebral cortex. And he defined a number of areas, which I will show you in some of the slides. And then he gave us the idea of the different types of cerebral cortex in the cerebral hemisphere. Before I proceed to the cerebral cortex, let me have a very broad outline of the cerebral cortex. See here, this is the hemisphere. This is cerebral hemisphere here. And you have the cerebellum. This is a medulla oblongata, pons, maybe somewhere here midbrain. Okay, this is the entire cerebral hemisphere. This is cerebellar cortex. Now, if you take the coronal section, this means if you cut it like this is the coronal section, and you get that, and you get this type of picture. What you see here, you have a gray matter, that is the neurons in the periphery. You just see that this gray matter is in the entire cortical element. The inside is the white matter. These are the fibers. These are the fibers. You can just see this is the white matter. Gray matter is outside, and white matter is inside. And if you are looking at the spinal cord, what is happening in the spinal cord? Okay, it's a small difference. That means in the spinal cord, the gray matter or the nuclei or the neurons are located inside, and the fibers from the neurons, they will be ascending in the dorsal column, the lateral column or interlateral column, and this is the anterior column. This is horn, dorsal horn, lateral horn, and ventral horn. So now this one, I come back here. That means outside we have the gray matter, inside we have the white matter. This cerebral cortical structure has been, it's not a smooth one. It has a very great number of foldings to increase the surface area to accommodate the neurons present. Now you have these, some of them, they are deep. These are those who are deep, they are called fissures, the central fissure. So you just see that this is a fissure. You have a central fissure or a central sulcus, and then lateral sulcus, something like that. And each of these gyri have a sulcuses. So that means the ups and downs. Downs are the sulcuses, ups are the gyri. So that is what the fissures are, the deeper in structures, they deep enter into this thing. So you can have one here and one there in the lateral side. You have in the central location the ventricles. Ventricles are filled with the cerebrospinal fluid. So that means they would nourish this the central core of the fibers. So you and the neurons present here, some of the neurons, the basal neurons are here. They are not, they are given faintly. So that is about the organization of the gray matter and white matter in the cerebral cortex versus the organization of the gray matter and white matter in the spinal cord. Now you just see that this is the great work of Konstantin von Economo. You see that he has mapped each and every millimeter or micrometer of the human cerebral cortex, and he has mapped and given the areas some name. It's a very, I believe it is one of those most difficult experiments. So now just briefing you, this is the frontal lobe, this is the occipital lobe, and this is the parietal lobe, and this is the temporal lobe. This is the lateral sulcus, this is the central sulcus. This is here the central sulcus. And if you look at this picture, and what happens, we will be whatever the nomenclatures economy has given, and the trouble here taken to give each one some segment or numbers, which are not easily understandable, which are not easily able to summarize. Okay, so now with that, then what the people did, or even economy himself, he divided the cortex after seeing so much complexity in the cerebral cortex. He divided the cerebral cortex into three major types. One, isocortex. That means iso is the neocortex, which has 6 layers. That means he has studied the layers of the cerebral cortex, and those who are those areas having isocortex are 6 layers. So then there are cortices. There is another extreme group, that is allocortex. That means they do not have 6 layers. They have only 3 layers. Especially these are the archie or the paleogenetically old cortical layers. They belong to uncus, the hippocampus, and the dentate gyrus. Okay, so these are allocortex, wherein 3 layers. So the iso is having 6 layers, allo is having 3 layers. In between, you have a mesocortex, that is in between this iso and allo, that is the mesocortex. Now, isocortex is the entire cortical mantle, which is shown in yellow here. This all makes the isocortex. This is frontal, the parietal. This is a sagittal section. That means you have cut the brain in between. Then you have the corpus callosum there, and then you have this part becomes the temporal component. This is occipital component. Frontal, parietal, temporal. Okay, so now you have this blue one makes the allocortex, the blue structure here. So blue structure is here, uncus is here, and then you have the hippocampus, dentate gyrus. Then it will be going on like this. This is corpus callosum. Okay, so now, and over this we have this cingulate gyrus. Over this corpus callosum, we have the cingulate gyrus shown in a deep yellow color, and the color which is mixed with the in between the light blue color and the light green color is olfactory. This is olfactory area. So now, what I want to say, this yellow one is the iso, 6 layers. The blue one, a straight blue one, is allo, 3 layers, uncus, hippocampus, and dentate. And these green and the pale structures, the deep yellow and the pale structures are the mesocortex. They form the cingulate cortex and the para-olfactory area. So these are the 3 major divisions the economo has made. Now, after seeing the 3 major divisions, let us see what this isocortex looks like. Now, the study was they have taken the cortical layers, put it in histology, used different staining methods. One, Golgi staining. Golgi staining is also known as a cellular staining. And this Golgi staining will demarcate the neurons and their fibers. You can just see that. This is Golgi staining. When you have stained with a Golgi, you have the neurons becoming clearly and their fibers are also coming up. So that means that's a Golgi staining. Then they used the Nissl staining. In this Nissl staining, the cressyl violet is used, and you have only it stains the RNA molecule. So that means it stains only the neurons. So that means the cell body is marked or and this also you will know the cell density. You can just see that how each one is stained, that it indicates the density. Then we have another staining, Wiegert staining. This Wiegert staining is done with the resorcinol and other compound, and this stains the myelin and elastic tissue. The myelin and elastic tissue, when it is stained, it only detects the fibers. Okay, with these three staining methodologies, they categorized into 6 layers in the isocortex. And these 6 layers are classical, and the cortical layers vary in the different areas of the brain, which I would deal subsequently. Now, these 6 layers are nomenclatured as 1 to 6 with a Roman letter. So the layer 1 is an external layer, the outside layer, and it is a molecular layer. Molecular layer. This is only small molecules are there, and this molecular layer are also known as a flexiform layer. This is also flexiform layer because you have the plexuses here. You just see that plexuses. So this is a molecular layer or a flexiform layer. That is layer 1. Then layer 2, you said that it is the external granular layer. The external granular layer, you have these granular type of cells there, and these granular type of cells, that is why it is called external granular layer, and you do not have much of these, and you have these granular here, the fibers are here and fibers are here, these fibers, horizontal fibers. Then third layer is a external pyramidal layer. So you just see that this is the pyramidal cells, the triangular cells. They are trying to send the dendrites upwards and the axons here in between. And these are external pyramidal cell layer. That is a layer 3. Layer 3. And you can see these pyramidal cell types. And again, you have these on the top of these things, there is a horizontal ramification. At the level of the base, there is a horizontal ramification. That is the external pyramidal layer. So then layer 4 is a granular layer. This is the layer 4. This is a granular layer, and this granular layer will have a large amount of the fibers horizontally spreading. But you have these granular cells. These are the inner granular layer, 4th one. Then fifth one is the pyramidal cell layer. This is the inner pyramidal cell layer or a giant pyramidal cell layer. So the very large giant pyramidal cells, large sized pyramidal cells are there. These are giant pyramidal cell layer. And sixth one is a fusiform layer. You have these fusiform cells. They are in the last layer. And below that is this part is only the white matter or the fibers. Okay, so now one more aspect here we have seen with the Golgi stain and the Nissl stain with these things. The Wiegert stain, you can see that these are horizontal myelin or these fibers going horizontally here, one band, the second band, third band, fourth band. And this is known as a Eichner's stripe that is going outside. This is the Eichner stripe, and this is a case Betreu's stripe. Then outer Billergar layer and inner Billergar layer. So these are nothing but the dendritic arborizations or dendritic projections. Okay, and even you see these cells, these inside, you can have these projections coming up and reaching at different layers that is seen. So that is the major broad cytoarchitecture of the cerebral cortex. So that is divided into 6 layers: the molecular layer, the external granular layer, the external pyramidal layer, the inner granular layer, inner pyramidal layer, fusiform layer. Just for your remembering, you have only molecular layer here, granular external, pyramidal external, granular internal, pyramidal internal, and fusiform layer. That is the 6 layers of the cerebral cortex. Okay, so now coming back with the outer layer, that is the external or the first layer, that is a molecular or a flexiform layer. Now, this is what I am trying to look at, the molecular layer or flexiform layer, and this is what is being rectangled with a red rectangle is the layer. They have the apical dendrites from the deeper layers. All these layers, they project on to the top, and then they ramify. The apical dendrites from these layers, these dendrites reach here. They ramify horizontally, forming a Exner layer. So this is a Exner layer. So they are just spreading horizontally. And there are very few neurons. You can just see that these nasal stain shows only few neurons. These neurons are neurons of what are called Cajal cells and Martinotti cells. Roman Y. Cajal is one of the great histologists, and he just using the silver staining, he mapped up the cerebellum very well, the Purkinje cells. But here, these cells are named after him, Cajal. Kajal, I'm not saying that Kajal, this in Italy, it's Cajal is a Cajal cells. And another type of cell is a Martinotti cells, or also known as horizontal cells here. They are very few. They are very few. It's only dendritic arborizations. Okay, the cells, the axon terminals of Martinotti, and you will see the Martinotti cells spreading the axon terminals there. Okay, so what are the functions of this layer, if you are looking at? So this layer is very important for our arousal response. They spread the positive or excitatory or inhibitory activity. Suppose if the inhibitory activity is more, so then the cortical activity is suppressed, or in excitatory activity is more, there is more activity. You may see different waveforms in the EEG. So that means that is so they are very, very important for our arousal response. That means they receive inputs from non-specific areas of the thalamus. So this one, though I have not mentioned here, they receive input from the non-specific areas of the thalamus, from the because there are specific areas of the thalamus, non-specific areas of the thalamus project onto this layer and spread the entire excitability and keeps us aroused. So this is arousal response. Output of these cells, they will reach to the within the cortical areas and opposite areas. The output of these areas, they will spread to the neighboring areas of the same cortex, or they will descend down and go to the other side. So that means the same or the other, but they are not many. Many of them are not going. So that is what the molecular layer is about. Now coming back here, the external granular layer. The external granular layer I have marked here. This is external granular layer here. The rectangle is moved down, and you have these granular cells. If you see that granular cells, they are closely packed. These are fusiform cells. Their dendrites reach layer 1, and 3. So that means so the layer 1 makes the Chi's, this one makes the Chi's stripe. So these horizontal stripes. Exon pass to exons of these layers. So these cells, granular cells, they go to layer 5 and layer 6. So they will descend down. They will go to 6 and 5. This is a 5 layer. So exons of these things. The neurons of this layer receive inputs from layer 4. So that means layer 4 would be communicating, not only layer 4, all the infragranular layer, they are all giving information to this layer. So that means layer Four in particular, but others are also giving the inputs. So they assist the signal processing at layer 4. They help or they try to discriminate, they try to modulate the signal processing at the layer 4, that granular cell layer. Output reach within the cortical areas and opposite areas. So within the cortical areas is known as a associative fibers. The opposite areas are known as a commissural fibers. So that means they have to pass through the corpus callosum and reach to the other cerebral hemisphere. So that means the output of this layer, they go within, that is associated fibers, and the other opposite side is the commissural fibers. Mostly these are association fibers. They will assist the inner granular layer, the inner granular layer. Now coming back with the third layer, the third layer is a external pyramidal layer. This is a pyramidal cells here. These are situated outside. These pyramidal cells, they have the dendrites. They are reaching the 2 and 3 layers. They will reach 3 and 2 and 3 layers, and their axons reach to the layer 5 and other areas. So let us see what that contains. The triangular cells with the dendrites reaching to 1 and 2. Triangular cells, dendrites reaching here. Basal dendrites reach within the layer 3. So the basal, these are basal dendrites. You just see this. These are basal dendrites. They are within layer 3. The efferents reach, that means axons of this, reach to layer 5 and layer 6. They will just come to layer 5 and they will go to layer 6. The efferents from this. The afferents from layers 4, 5 and 6. It receives inputs from 4, 5 and 6. That means from granular cell layer and infragranular layers, all the layers, bottom. The output from these cells reach within the cortical hemisphere, that is associative fibers, and the opposite side as a commissural fibers. Okay, that is about the external pyramidal layer. Now we have the major cell layer. This is the inner granular layer. Usually the cerebral cortex is recognized by some workers. They believe, or functionally it can be divided into granular layer, supragranular layer and infragranular layer. Granular layer is the demarcation point. This is an important layer. So densely packed small star-shaped or stellate neurons. You just see that stellate neurons there, small star neurons. The more the neurons, they become dust-like. That is called as a coniocortex. Conio means dust. The dendrites ramify horizontally within the layer. So the dendrites here, dendrites of these things, you can just see that these dendrites are here like this. You can just see that what I have made yellow. Below the yellow, you have these horizontal myelins, fibers going. The dendrites reach within the layer, and this is known as a external layer of bilar core. This is the external layer of the bilar core. These receive inputs from specific areas in the thalamus, from the specific nuclei of the thalamus. Say, for example, I said ventro-posterior-lateral nucleus of the thalamus. Yes, they will relay in the parietal cortex, that is the postcentral gyrus, area 312, here in this granular layer. So that is one specific projection. Okay, suppose if it is a visual cortex, that is released into the lateral geniculate body. If it is an auditory cortex, lateral geniculate body is a structure of the thalamus. And if it is an auditory cortex, it is relayed into the medial geniculate body. Like that, each of these, if it is coming from the basal ganglia, they will go to the anterior thalamus or lateral anterior thalamus, interlateral or lateral thalamus. So then if it is with a limbic system, that may be with the medial part of the thalamus. So like that, there are specified areas of the thalamic nuclei. They reach to this layer 4. That means they receive inputs from the thalamocortical projections. They are specific. The non-specific one, I said, the layer 1 receives the non-specific areas of the thalamus, non-specific nuclei of the thalamus. I will deal with them when I talk about the thalamus, and you just have the idea about that. The axons of the stellate cells reach to the layer 5 and the layer 6. The axons from these cells, they will reach to layer 5 and layer 6. This is layer 5, the neighbor, the just immediate neighbor, and the layer 6. What is the function of this layer? This receives sensory input, tactile localization. That means the localizing the particular, yesterday I was talking about the sensory homunculus, that topographically they are arranged. The finger area, in the finger area, I have a cerebral cortex, the finger area that is represented here. So they are for the localization of the particular area, dermatome area, and it will also identify the intensity of the stimulus or sensory modality, and also it will map, or it will try to give the pattern of the mapping, whether it is a touch, pressure, or pain, whatever is coming. So these are the functions of these layer 4. This is an important layer. So then this is, I would say, this is a sensory layer. As far as me is concerned, this processes the sensory information. So then we have another important layer. This processes the motor information. That is the all the motor activity is governed by this. This is a inner giant or a large pyramidal cell layer. This is a layer 5, is a giant pyramidal cell layer or a pyramidal cell layer. These are pyramidal cells. They contain large or giant pyramidal cells, 60 to 120 micrometers in size, and their dendrites reach all layers above, supragranular layers, ramify, they just spread out. So apical dendrites reach all layers above, and the basal dendrites ramify horizontally. So basal layers, they ramify horizontally. So you just see that. And these ramifications, they make the inner layer of biliary cord, inner layer of biliary cord. So that is the inner biliary cord stripe. That is what they show. Okay, the inputs for this layer are from layer 4 and layer 3. So they receive inputs from the layer 3 and the layer 4. That means all sensory modalities. Suppose if the sensory modality is there, there may be an action required. They will excite the motor neurons to see that a particular response is made. The response is made in terms of motor activity. So they are receiving input from the layer 4, that is sensory, and the layer 3 is also a part of the sensory. So the assisting the externopyramidal layer. So that means they receive because these layer 3 receiving a commissural fibers from the other side. Okay, axons of these giant pyramidal cells, these axons, these are very long axons. These long axons reach to the spinal cord. Maybe you just see the cerebral cortex here. You have the sacral segment down the lower part of the vertebral column. So now they have such a long axons from that to this thing. So they reach the brainstem and the spinal cord. So that means they will reach all the motor neurons, which are these are called what are called the upper motor neurons. They reach the brainstem and the spinal cord. Where in the spinal cord we have the lower motor neuron, that is alpha motor neuron, which reach to the muscle. And this lower motor neuron, they will descend down and cross and then approach to the spinal tracts. So that means these are long axons. They will reach to the spinal cord and brainstem. And their function is these are motor executive functions. They perform they bring in action and various parts of the brain, and the spinal cord. Even they bring in action of the, they produce actions of excitability in the reticular system, the maybe in other parts of the brain. So that is, these are long axons. Now comes the sixth layer, that is the fusiform or multiform layer. So you just see that this is a fusiform or a multiform is a different type, varying type, and these are fusiform cells. Dendrites reach the outer layers. The dendrites, all these outer layers, they will receive inputs from fourth and fifth. So from fourth layer and the fifth layer, they give inputs here. They give inputs. Axons of the satellite, axons of the stellate cells reach up to layer. These cells, they will reach to this, these axons, they will reach to this thing, layer 5 and other cortical areas. That means by the side and by the opposite. The sensory processing, they help in the sensory processing, also the motor processing, and spreading impulses to the subcortical structures and also to the opposite side. That means both have associative and commissural fibers, mostly associative. So this is in brief about the various areas of the cerebral cortex. Now, coming back here, just whatever I have mentioned, I again try to fill in certain information which may be bothering you or which may be essential to remember. What are the afferent connections of the cerebral cortex? It's a simple question. The afferent connections of the cerebral cortex. If you have a question like what are the various afferent and efferent connections of the cerebral cortex, and mention all the layers of the cerebral cortex, how they are connected. So in that case, I'm just trying to give you certain information. So this, number one, receives specific thalamocortical projections. Especially these thalamocortical projections reach layer 4. That is inner granular layer. And layer 3, it is a external pyramidal cell layer. And this would give a collateral to the layer 5, that is the pyramidal cell layer. And these specific connections, typical examples are the somatosensory area 1 or the visual association area visual area number 1, what is called a V1, or we can have the auditory area number 1, A1. Non-specific thalamocortical projections, that means they do not have the specific nuclei in the thalamus. They reach layer 1 and collaterals to each and every layer, layer 2 to layer 4. There are the afferent connections, the association fibers, that means coming from the other areas of the brain, connecting different areas of the cerebral cortex. That's the association fibers. That is the inputs they receive. The inputs, they come to the layer 1, 2, and 3. That means at each level. Commissural fibers, that is coming from the opposite hemisphere. And these association fibers terminate in layer 1 to 4. That is what the afferent connection is. Okay, so now the efferent connections. Here I have taken this from Jones 1995. You can just see the different neurons have been made. The D is this is the inner outer granular layer. The number 2, the 1, 2, 3, 4, 5, 6. This is outer granular layer. So this is the neuron here, and this is a neuron. You can just see the axon going. So what type of axon it is giving? Cortico-cortical. So that means the associative fibers. So then we have the number C here. What is this? This is coming in layer 3. Layer 3. That means outer pyramidal cell layer or external pyramidal cell layer. And this external pyramidal cell layer, where does it give? This gives the output to the corticocortical, that is associative, and commissural, so that means opposite side. So it gives the inputs, it gives outputs to the opposite side, broadly opposite side. This is on the same side, this is to the opposite side. These are commissural fibers. They connect the other hemisphere with this hemisphere. So that means what is happening, what right brain is thinking, the left brain should also think the same, so that to bring about the aspect, though they may be different in performing the actions in a different way. So then we see another this is a layer 5, the giant pyramidal cell layer. You can just see that. In this giant pyramidal cell layer, what's happening? This output goes to subcortical structures. These outputs, this goes to subcortical structures. Some are corticocortical. So corticocortical is in this way. So then we have the layer 6, layer 5 and a layer 6. They are layer 5 and layer 6. They will go to corticothalamic, corticoclaustral, and corticocortical, and some this thing. Here in this layer will go to the corticospinal, corticobulbar fibers. These are corticospinal and corticobulbar fibers. Here these are corticothalamic, corticoclaustral, that is a colostrum, that is a commissural, and then corticocortical, so within the association areas, and crossing. So these are the efferent connections of the cerebral cortex. If I were to say that efferent connections are given to the area, adjacent areas, the opposite areas, then to the spinal cord and brainstem, and to thalamus, the claustrum, the other parts of the cerebral hemisphere. So the entire cerebral hemisphere. Say for example, you have a what is called a Wernicke area. So this Wernicke's area is a speech center. So from this, the fibers reach to the Broca's area, that is the motor component of the Broca's area. These type of connections, these whatever the connections, these are coming here from layer 6. Now, the efferent connections of the cerebral cortex, they are originating from the efferents come from layer 5 and layer 6, the infragranular area. They are projection fibers, projection to the spinal cord, projection to the brainstem, projection to the other areas. And the commissural fibers, opposite hemisphere, association fibers within the hemisphere. The pyramidal and granular cells of 2 and 4, they give inputs or the efferents to the 2 and 4 layers of the same cortical areas. So that means they will try to modulate the output of these because they receive inputs from the layer 4. Then Martinotti and horizontal cells of the same. This is layer 1. The Martinotti and horizontal cells, they are mostly present in the layer 1 and 2. They will go in the same areas. That is the efferent connection. Now, just to summarize the functional layers of the cerebral cortex, I just try to make the, I was trying to impress upon you in between that layer 4 is the demarcating layer. That is a granular layer. That is a layer 4. Then anything above or outside the layer 4 is a supragranular layer, that is layer 1 to 3. Anything below the granular cell layer, that is a infragranular layer. These are just, I want to remember in that way. So there are 3 layers above the granular cell layer. There are 2 layers below the granular layer. So that means broadly, these are functional areas of the cerebral cortex. Now, what are those supragranular layers? If I am trying to look at that, layers 1 to 3, these layers are primary origin and a termination of a intracortical associational areas. I have already mentioned about that, and other areas of the hemisphere. Intercortical commissural connections, the connections between the cortex to the opposite hemisphere through the corpus callosum. It is very well developed. Supragranular layer is well developed in humans, permits communication between various cortical areas. Now, granular cell layer, this granular cell layer is layer 4. That is the inner granular layer. Receives the thalamocortical connections from a specific thalamic nuclei, very clear. And it is a sensory cortex, prominent in the primary sensory cortex. Especially it is so prominent in the visual cortex and auditory cortex that it dominates in all the layers of the cortical, all the cortical layers. Now comes the infragranular layers. Infragranular layers are layer 5 and layer 6, connects cortex and subcortical areas. These are very well developed in the motor cortex, the motor cortical areas. And these are more developed, especially the special motor functions. In that case, they are only present. Other cells are been either absent or they have been occupied. That is what I'm trying to talk about here. Agranular cortex. That means there are no granular cell. It's extremely small or non-existent granular layer 4. Layer 5 give rise to principal cortical efferent projections to basal ganglia, brainstem, and the spinal cord, and to the cerebellum. That is through the brainstem. Layer 6, a multiform or a Flexiform layer projects to primarily to thalamus. This is a brief about 3 components, 3 functional components of the cerebral cortex. Now coming back here, the different types of cortex according to Economo. I just bring back that old picture. So this, the yellow one is the isocortex, having a 6 layers, but all the 6 layers are not alike. So then this intermediate connections, this color you can just see, this is a mesocortex that is between allo. This is allo is 3 layers, iso is 6 layers. It is between allo and iso. And these are present in this is cingulate cortex. This is cingulate cortex here. And the para-olfactory area, this is olfactory area, this is para-olfactory area. So now allocortex is this blue one. That's why I have made the blue coding. This is uncus here. This forms the uncus. Then hippocampus. This is hippocampus, dentate gyrus, and then part of the projection of the dentate gyrus. So then that is three different layers. Now, how these isocortex? What I was trying to impress here, not all the layers of the isocortex are similar, because this is sensory, this is motor, this is motor, this is sensory, and somewhere here it is different. So now looking at that, it is being divided into 5 different types. 5 different types, depending upon the cell type. Here, this one, the layer the one agranular cortex. This is the agranular cortex. What you see here in this agranular cortex, you see only pyramidal cells. Pyramidal cells. You see 1, 2, 3. 3 is A, B, C. You can see that layer 3 has become more. 4 is almost absent. It's not there. 5th is very well developed. 6th is also Flexiform is developed. So this is a motor cortex, especially present in the posterior third part of the frontal lobe. So posterior third of the frontal lobe, and especially Broca's areas, these have this type of agranular cortex. Now, in reference to this, we have a granular cortex. You just see this. This is a granular cortex. Here in this, it's only granular layer. Granular layer is developed, and you see that the layer 4 becomes ABC. Layer 4 here 3 becomes ABC. Layer 4 becomes layer 4 is a granular cell layer. Layer 4 becomes thick, the ABC, and they will have that analysis of the visual area. This is more so in case of the V1 and the auditory cortices. This is a granular layer. This is sensory cortex. This is a motor cortex. So then frontal cortex. Frontal cortex, it has both the sensory and motor mixed together. That forms the classical 6 layers. Classical 6 layers. Frontal cortex that is representing the anterior 2/3 of the frontal lobe. So then parietal cortex, again parietal cortex is the dominance of the granular cells because it is a sensory cortex. It has a sensory cortex. You have this layer 4 is a little more evolved. Layer 4 is a little more evolved. So then polar areas, especially the frontal and the occipital polar areas, they have these two regions. All the cells are compressed. They are having the representation, but they are compressed. They are having a smaller areas. That is what I have mentioned here. A granular is the motor cortex. The number frontal is the anterior 2/3 of the frontal cortex. Then parietal is the parietal, occipital, and temporal areas that constitutes the sensory cortex. This is the parietal cortex, and polar is the on either pole of the cerebral hemisphere. They are all compressed. You see that the layers are there, but they are compressed. And granular is only granular complex, especially present in calcarine. Calcarine cortex is the visual cortex. Visual area, primary visual area, V1, or Hessel's gyrus, that is auditory cortex, that's A1. Okay, having seen these different types of isocortex, we look into the mesocortex. So now I have mentioned about the mesocortex. This is one of the mesocortex here. You just see that anterior cingulate sulcus. In this anterior cingulate sulcus, the area 24C, I will just mention about that. This is the Brodmann area 24C. What is happening? Layer 1 is present, layer 2 is present, layer 3 is present, layer 5 is present, the layer 6 is present. This is white matter. So there is a missing layer 4. Similarly, you have different combinations, combination of these layers. Some of these layers either absent or they are having a little occupancy. So that type is a mesocortex. These are some of the mesocortices which I have shown here. Especially this one is remarkable. Now, what is allocortex? Allocortex, I have these represent the hippocampus, uncus, and prepiriform cortex. So there are 3 layers. The pyramidal cell layer, this is the pyramidal cell layer, then outer molecular layer, and inner polymorphic layer. That is what happens in case of hippocampus. In contrast, you see what is happening in the isocortex. You have all these classical 6 layers. The molecular layer, the outer granular layer, the outer pyramidal layer, the inner granular layer, inner pyramidal layer, inner fusiform layer. So just I am repeating just to make you feel that isocortex is having all these 6 layers. Here there is only 3 layers, the central pyramidal layer and the molecular layer externally and a polymorphic layer internally. So this is allocortex. Now to summarize, we have 6 layers. I repeat, and these 6 layers are defined as for the Golgi staining, that is a silver staining, or a crescile blue or crescile violet staining, that is only cell types, or Wiegert staining for myelin sheath. So they classified into 6 layers. That is a classical standard isocortex. That is that forms the 90% of the entire cerebral hemisphere. And this outer molecular layer, the outer granular layer, outer pyramidal cell layer, the inner granular layer, and inner pyramidal cell layer, and a fusiform cell layer. Here I have mentioned about the molecular layer. It is the function is arbusal. The external granular layer is associated, that is, it's connecting the neighboring cortices. The external pyramidal cell layer is the associative as well as commissural. It talks to the opposite hemisphere. Then the inner layer is sensory. The inner pyramidal cell layer is motor. Inner granular layer is sensory. The inner pyramidal cell layer is motor. And fusiform layer is a commissural and associative. It talks to thalamus and connects to the entire cerebral hemisphere in one as well. Okay, I'm trying to provide you in this whatever the lecture, I try to provide you the cytoarchitectural areas of the cerebral cortex may be useful, because whenever we talk or whenever we try to represent some neurophysiological data or neurophysiological discussions, we come across these things. I thought I should deal with you. Now, the major books here, I've taken it from Samson Wright, and I've taken it from the Candles and the Guyton and Genong. They have a very little information. Whatever the information I have given is nearly 70 to 80% is present in Samson Wright, and in the remaining part I have taken it from Candle. And maybe entire whole thing becomes the good part for you to understand the neurophysiology. Okay, so now the assignments I am trying to give you here, because there are so many terminologies I have used. First, draw a neat label diagram of a typical isocortex. Please make an attempt. Give details of the connections and functions of each layer. Perhaps what I would do is I would draw this and I would write these whatever I have written. So you try to go back to my lecture and try to bring this, you can have the entire thing summarized. Now, write short notes on outer molecular layer. I have described each one of them. The commissural fibers, giant pyramidal cell layer, the sensory layer of the cortex, that is the inner granular layer, the functions of the inner fusiform layer, isocortex types, that 5 types I have mentioned. You write all the 5 types. Allocortex, you define that. Then brief answers, functions of various layers of the cerebral cortex. You answer them briefly. Draw label diagram of the typical isocortex. Again, the same thing you practice. Cortical rays receiving sensory input. It should be layer 4. Then cortical layer giving efference to the spinal cord. It should be layer 5. Commissural fibers, supragranular layer, associative layer, infragranular layers, the layer involved in arousal response. So I have given you all type. You just like to look into these particular assignments, and if you can do it, and you have a full control of the entire cerebral cortical organization. I will be dealing in the next class the cortical areas and their functions. Okay, thank you.